Hello, good evening. Good evening, Abhirup. Just hold on for a second. I'll begin after 2 minutes. Hold on. Okay, so it's not 7 yet. Okay. Good evening, Suraksha. So today we will begin with the chapter photosynthesis. Photosynthesis. So this is the chapter we will begin with. Okay, so it's already about to be 7. I'll wait for 1 more minute, and right to the fourth minute of the video I will begin teaching. Okay. So you're almost there. So let's begin our understanding about photosynthesis. See, okay, so let's begin our understanding of photosynthesis. I hope you can see everything clearly. Okay. So is this everything clearly visible? I hope everything is clearly visible. Okay, I hope this light is the best one. Or maybe okay. Hello, Snehanshu. Okay, I think this is the best light. So now, moving forward with our understanding of photosynthesis, you know that photosynthesis is what? Is the process of making food. Is the process of synthesis of C6H12O6, that is glucose, and glucose is stored as starch in plants. We know this, right? That now photosynthesis is the process by which green plants, or organisms, better to say in class 11 level, organisms containing organisms containing photosynthetic pigments. Now by photosynthetic pigments, organisms containing photosynthetic pigment, I also mean algae and photosynthetic bacteria. So these are the organisms, which carry the photosynthetic pigment. They can do photosynthesis. They can do photosynthesis means they can prepare C6H12O6 in higher plants In higher plants, the glucose is stored as starch. Multiple units of glucose, when forms a polysaccharide, then it becomes starch. Many students ask me question that why not store it as glucose? You know that in animals or in humans, the extra glucose that we have taken up is not utilized to form energy, is stored as glycogen. Again, it is a polysaccharide. And glucose is a monosaccharide. Fine. So there is a monosaccharide, there is a polysaccharide, and as you can see that always the food is not stored in the simpler monosaccharide form, but they are formed in a complex polysaccharide form in plants and animals or humans, as per that matters in this example. Now question arises that why is it so? Why not stored as glucose? Why not stored as glucose? Why we need to store it in the polysaccharide form? Because if you have stored as glucose, then it would have disbalanced the osmotic concentration inside and outside the cell, which would result in bursting of the cell, and hence it is always stored in a polysaccharide form. Now this is the reason. However, why do we take food? We take food, or we take any food, we convert it into glucose. As we know, we take any food, we do convert it into glucose, and when we convert it into glucose, that glucose is broken down to give ATP, which is known as respiration. Now that ATP is produced with that CO2, and H2O is also released in a normal respiratory process or respiration. However, plant, we take this glucose from external source, or any food material as for that matters from external source, and hence that food material, be it a protein source or a lipid source, whatever, or be it a plant source, it's converted into glucose, and then we utilize our nutrition by the process of respiration, which is breaking down of glucose into CO2 and H2O. Hence ATP would be released. So that thing we know, and we understand that thing very properly. Okay, but in plants or in photosynthetic organisms, if I say, in photosynthetic organisms, raw materials are utilized. Raw materials are utilized to make up the food source. Means they make their own food, like the kids say. So which are these raw materials? These raw materials are basically CO2 present in the atmosphere, as you know, and the H2O from the soil, and both of them combine to form C6H12O6. And for this they require a lot of ATP. They utilize the ATP, and also they utilize another thing, that is sunlight. And also there has to be the presence of photosynthetic pigment in that bacteria. So number one, CO2 is required. Number two, H2O is required. Wait, hold on for a second. Number one, CO2 is required. Number two, H2O is required. Number three, chlorophyll is required. And number four thing that is required is your sunlight. Now this chapter will deal with a detailing of the chlorophyll or photosynthetic pigments, or sorry, I think it was not visible. See, number one, CO2 is required. Number two, H2O is required. Number three, chlorophyll is required. Number four, sunlight is required. Chlorophyll, we can say photosynthetic pigment. Okay, so I'm sorry, I did not notice this thing also. Okay, so here we know, and in this chapter we will understand the detailed optimization of the process, that how the process occurs, and everything. So So slowly we will move forward and understand that what is the function of the photosynthetic pigments. We will understand that what is the function of these four raw materials and how they are utilized and from where they are utilized. And also we will learn about photorespiration, which is a slight modification of the photosynthetic process. Slight modification means there is modification in the chemical reaction part of the photosynthesis. Now if we want to understand a photosynthesis, okay, already class 10's knowledge is there with us. But however, before we go into and understand the entire photosynthesis process that I am depicting here, hold on for a second. Okay, so like if I have to understand the entire photosynthetic process that I will try to depict here, let us go into some historical perspective of photosynthesis. So by historical perspective, I try to mean that the experiments which helped us to understand the photosynthetic process. So okay. So by historical perspective, if we go into the historical perspective of photosynthesis. So in historical perspective of photosynthesis or history of discovery of photosynthesis, now which is important to understand. Now many students complain me or many students say that what is the requirement of understanding the history? If you don't understand how things were discovered, you will never understand the importance of the things. Okay. So first scientist that we will name off is number one. First scientist we will name off is number one, Van Helmont. Van Helmont. So here you find out, okay, here I have not noted down, so here you find out. You see, Van Helmont, what he concluded is that food of plants comes from water and not from soil. So this is his conclusion, that food from water actually comes from, food for plants actually comes from water. Other people, please check if it is clear or not clear. Please check if it is clear or not. Are my writings clear? Okay, Naina, please, I think you should check your internet connection little bit, beta. Okay, so food. Now, what Van Helmont did conclude, Van Helmont concluded that food from water comes from food for plants comes from water and not from soil. Actually, what is the experiment that he did was that, suppose he took a pot, okay, and he weighed some soil and put the soil inside, okay, and he allowed the plant to grow. Here he put a sapling, okay, and he allowed the plant to grow, and the soil was there. After a certain day of time or a certain years of time, when he again took out the soil from there and measured it, he found that the mass of soil has decreased by a very less amount, but when he measured the plant, first he measured the sapling and later on he measured the plant and said that the weight of the plant or mass of the plant increased by many folds. Suppose, for example, let us consider that at first the sapling, just the sapling was of 10 gram. Later on he saw the sapling had become of 500 gram. And the plant has become of 500 gram. And he saw that the soil is, suppose at first was of 50 gram. Later on it became 49.5 gram. So the decrease in the soil value is 0.5 gram. However, the increase in the mass of the tissue mass of the entire plant is around 490 gram. So doesn't really calculate, okay? So if plant would be taking all its food material, from the soil, okay, if plant would be taking all its food material from the soil, then what would you conclude? That the weight of the soil would have gone down by that proportion only? But it did not happen. It did not happen. The weight of the plant remained, increased by many fold, but the weight of the soil did not decrease by that many, folds or that many proportion. So he concluded that it is probably that he told that it is probably, I'm sorry for the message tones. So he said that it is probably because that the entire tissue mass must have come from something else. Now he couldn't conclude. I'm so sorry. Okay, so I hope that no more messages will come. So now what he concluded is that okay, fine, if soil is not providing for the almost all the tissue mass, then there must be something. He couldn't get the idea that it is actually CO2 from the atmosphere that is contributing to C6H12O6, which is contributing to the nutrients or nutrition of the plant, which is helping us to build helping the plant to build its body or build its tissue mass. Agreed? Okay, so I hope that you have understood that he first actually took away. Now why is it important, sir? Why knowing his experiment is important? Knowing his experiment is important because it actually concluded that okay, fine, it is not from soil. At least this conclusion people came to know, and which generated the thought that if it is not from soil, then from where? So that's why it is very important to understand. Okay, now second experiment is by Joseph Presley. So the same trigger, and Joseph Presley was right at the point to explain the next thing. Okay, so the second was by Joseph Presley. Now in Joseph Presley's experiment, he tried to figure out, okay fine, if not soil then what? Only water it cannot be. Okay, so he tried to figure out. Now he couldn't figure out the entire thing, but what he told is he performed an experiment. He put a bell jar on a burning candle and he put a bell jar on a plant, and he put a bell jar on a plant. Okay, so this he did, and this is 1, this is 2. Again what he did in the third thing, he put the candle and the sapling or the plant in the same bell jar. Now if this was the situation, he saw that the plant here died very early. The candle also, got extinguished early. But here the candle continued to glow and the plant survived for longer period of time. And the plant survived for a longer period of time. Same experiment he repeated with rat. He took a bell jar, put a plant and a rat. Suppose this is rat, which is actually a breathing animal here. Okay, together. And then he put the rat and the sapling together separately also in the same way. But he found that when he put the rat, here imagine the rat and the sapling separately, the rat also died, the plant also died early. But however, when together the rat and the plant stayed in the same bell jar, then what happened is that they both remain alive for a longer period of time. This made him conclude that what? This made him conclude that the photosynthetic organism or the photosynthetic plant restored the photosynthetic organism or the photosynthetic plant, or the plant restores to the air whatever a breathing animal or a burning candle removes. Now he didn't know much that what is taken, what is restored. So we today from our perspective know that plants by the process of photosynthesis give out O2, which is utilized by a burning candle for burning or by a breathing organism for respiration, and they in turn give out CO2, which is utilized by plants. So this we know today, but at that time Joseph Presley didn't know, and they were after Van Helmont's experiment that no, it is not from soil, like you have seen that no, it is not from soil. Then after Van Helmont's experiment, they were at a searching thing that they were searching what it is. Joseph Presley tried to conclude that okay, it is from atmosphere, but he didn't know where it is from atmosphere. Okay, but he didn't know that which gases are restored by plants and utilized, and again they produce something which is utilized by plants. So he just simply said, a much simpler version, he just simply said at the beginning that the photosynthetic organism or the plant restores in the air whatever the breathing animal or the burning candle removes. So this is the basic thing. Okay, now moving forward, then there is experiment of number 3, Engelmann. These German people have very difficult names. Okay, so anyway, Engelmann, this was the scientist. He said that sunlight is essential for photosynthesis. He said that sunlight is essential for photosynthesis, and he also experimented with, and I hope that I don't need to say you that what is the experiment that says sunlight is essential for photosynthesis. You have very well learned in class 10 the experiment that sunlight is essential for photosynthesis, right? if you put a dark cover, this thing, dark cover, then here starch is not present. So all these things were there. So yeah, so I hope that separately what experiment he did, we don't have to know right now. But yes, sunlight is essential for photosynthesis. That was said by Ingenhouse. Now he also experimented on aquatic plants and first time said that there is some gas emitted out or evolved out, sorry, better word, evolved out during photosynthesis. Now what is the gas that is photosynthesis? Now we know it to be O2. But his bubbling experiment, the bubbles proved that yes, a gas comes out. So look at these three experiments. First of all, a person says that okay, that its soil is not the substance which gives nutrition or the entire tissue mass or growth or food to the plant. I have experimented and I said you, if it's not soil, then from where is the mass or the nutrition coming? Joseph Presley tried to say, okay, it's from atmosphere, because I have experimented that whatever is restored in the atmosphere by the burning candle of the breathing organism is taken up by the plants, and the plants restore something which helps them to keep alive, and both support each other. So some thing is in the atmosphere, but he didn't know what. But we know today it's CO2 and O2. And look at the third experiment. They didn't know what. Joseph Presley didn't know what. Okay, but then Ingenhouse told that okay, it's some gas. It is some gas which is evolved out. So you know they started providing links to each other. That okay, fine. So this is what is happening to the process of photosynthesis. Now moving forward, number 4 is the experiment of Sachs. SACH, Sachs. Now he said plant produce glucose during photosynthesis. Number 1. He said plants produce, hold on for a second. He said that plants produce glucose during photosynthesis. Plants produce glucose during photosynthesis. Okay, so this was said by Sachs. Okay, fine. One more landmark discovery. 5th landmark discovery was by Engelmann. Engelmann. 4th discovery was by Engelmann, and his experiment was with green algae. There is a green algae, Cladophora. Now his experiment is a very important experiment that I must say you. Okay, what was his experiment? Actually what he did is, I'll take the next page to explain you. Just hold on for a second. Thank you for your patience. Okay, so next is the experiment done by what we can say is Engelmann. Now his experiment was that he passed light through prism. When he passed light through prism, then you very well know that you will obtain the VIBGYOR or the spectrum of light, right? Spectrum of visible light. Now when you obtain the spectrum of the visible light, so what you do is you take a tube and uniformly you distribute Cladophora. The Cladophora algae is distributed uniformly in this tube. it was seen that at the blue region there was more deposition of cladophora, and at the red region there was more deposition of the cladophora. So from here we can conclude that as because there is more deposition or more growth, definitely here photosynthesis is more. That means at red region and blue region, at red region and blue region or wavelengths of visible light, the photosynthesis is most effective, okay, or most productive. So definitely a factor of the wavelength came into existence right after the explanation of what we can say Engelmann. So Engelmann did a really great good experiment, which was really very important for us to understand. Now next is by Van Niel. Sixth experiment is by Van Niel. Now he experimented with purple and green bacteria. He experimented on purple and green bacteria. Now listen, this is interesting. He started experimenting on purple and green bacteria, and he also said that photosynthesis is light dependent. Without light, he also said, this was also said by Eisenhower, and he also said that photosynthesis is light dependent. Now his experiments on purple and green bacteria means there are type of bacteria or photosynthetic bacteria which are known as purple sulfur bacteria and green sulfur bacteria. Now I will talk about the details of purple sulfur and green sulfur bacteria and why they are called sulfur. But before that, you have to understand in purple sulfur bacteria there is no green pigment, and in green sulfur bacteria there is green pigment. But still, both of them does photosynthesis. So it was his discovery who said that you just need a pigment for capture of color. Maybe the rate of photosynthesis may vary. Maybe due to the color of the pigment, some pigment may be more effective or more efficient in doing photosynthesis, some may be little bit less efficient in doing the photosynthesis. But however, with the presence of light, there should be also presence of photosynthetic pigment. Now photosynthesis is a light dependent reaction, we say. He also said, the third point that he said is that H2 from suitable oxidizing agent is taken up to create reduction of CO2. Now that H2 can come from H2O. Now to make you understand that what is the importance of this finding, that H2 from or hydrogen from suitable oxidizing agent is taken up to create reduction of CO2, I will discuss little bit, and then you will understand that what is the importance of this statement. Let us go into it. Look, In photosynthesis, what happens is that the plant takes CO2, or any photosynthetic organisms take CO2 from the atmosphere and also H2O and from the soil, and hence both of them combine. Actually the H2O ka O goes out, O2 goes out, and H2 is taken up and it combines with CO2 to give C6H12O6. That is, the C of the glucose comes from the C of the carbon dioxide. The H comes from the water, which is actually a oxidizing, a reducing agent. I am so sorry. Good reducing agent, not oxidizing agent, reducing agent. Okay, so H2O is a good reducing agent from which H comes and they combine. In place of H2O, you can use an H2S also. Who uses it? Green sulfur bacteria. That's why they are called sulfur. Green sulfur bacteria and purple sulfur bacteria. What they do is they use H2S and combine them with CO2, and when the H2 is taken from this reducing agent and CO combines with CO2 to give C6H12O6, the O is coming from here only, CO2 ka O only, and this sulfur S, as the granules of sulfur are found, or these granules of S are found in green sulfur and purple sulfur bacteria, which is quite interesting to know. Now, I hope that you understand that what he tried to mean is that H2 from suitable reducing agent is taken up to create reduction of CO2. So that is also one of the path-breaking landmark that should be understood, that yes, this is also an experiment which gave us insight of photosynthesis. Now, 8th experiment was performed by whom? Okay, so let's see whom we can pick up. Robert Hill. Robert Hill. Now what happens is that plants use light energy to generate reducing power. Plants use light energy to generate reducing power. Wow! Just now in the previous experiment you have learned that okay, CO2 plus H2O, and H2 is broken down. H2 is the reducing agent from where H2 comes out, or H comes out and combines with CO2, but this process is not so simple. H2 is broken down with the help of light, which is called photolysis of light. Photolysis of water, sorry. The photolysis, breaking down of water, and hence the hydrogen comes out and combines with oxygen. Combines with carbon dioxide. Now this reducing power is generated by whom? By light. Hence this photolysis of water is also known as Hill's reaction. So we will learn about the details of Hill's reaction also. I am saying this in a very simple way. These things are not so simple when I chemically go into it. But yes, this is the overall look. Now, see, there is a confusion. H2 is used, or H2S is used, or any other reducing agent, or any other reducing agent giving H2 is used. What is used? Then come a scientist, number 9, this question. Theodore de Saussure, I think he is Japanese. Okay, he said that H2O is essential in photosynthesis. So that means it got confirmed, okay, so the reducing agent is H2O due to this scientist. Then came the 10th, 8th, then 9th. Oh, 7th, 8th, then 9th. Then came Emerson, another scientist, Emerson. Let's see what Emerson said. Okay, so we will try to understand what Emerson said. That there exist two photosystems. Now we will talk about the details of photosystem. There are existing two photosystems, photosystem 1 and photosystem 2, which are actually I can say which are assembly of photosystems are assembly of photosynthetic pigments. There is not just one kind of photosynthetic pigment, but a lot of kind of photosynthetic pigment in a higher plant, and they are assembly of photosynthetic pigment, and they exist in two assemblies, photosystem 1 and photosystem 2. And this was said by Emerson. Emerson gave the Emerson's effect, which we'll learn in detail in our coming classes. Emerson's effect, which we'll learn in detail in our coming classes. And this Emerson's effect gave us an idea of red drop, which also gave us idea of photosynthesis. Detail of this is more important, and we'll go into it very soon. Okay, and then Emerson also said that, hold on for a second. Let's say that there exists both light reaction and dark reaction. There is existence of both light reaction and dark reaction in photosynthesis. So that is the thing. Now moving into the last or 11th discovery is the Kelvin cycle. Sir, what is Kelvin cycle? Kelvin cycle was discovered by three scientists, Kelvin, Benson, Besham and Kelvin. These three scientists said that okay, CO2 is converted to C6H12O6. How? Through a series of chemical reactions. Through a series of chemical reactions. And hence that is known as your Kelvin cycle, which is literally the dark reaction. Now we know. Look, if you look into any refresher book or any other sites or anywhere, you will at least find 50 to 60 scientists' name, this, that. I have only told you the important ones, which is essential for understanding the process of photosynthesis. So having a look on the overall thing of today, what I have said you is this thing. Have a look on it. Take a screenshot if you want, because later on you have to make note, then you don't have to see the video again. You can directly make the note. And if I move up, this is what I have taught. And then this. Why is the name of Kelvin only used for the cycle and not all the three scientists? Snehanshu, very good question. I am answering you this. So then this. And then this Joseph Presley experiment. This was Van Helmont's. Then there was this. And then there was this. Ingenhouse and Sachs experiment, then Engelmann. This experiment was pretty interesting, which told us about which range of light is maximum effective in photosynthesis. Then there was Van Niel, who told us a lot of things about the purple bacteria, sulfur bacteria and the reducing agent and everything. And then there was Hill's reaction. This is I used you to make you understand the Van Niel's experiment. And then there was Robert Hill, breaking down of water, photolysis of water, Theodore de Saussure, and then there was Emerson, and then there was Kelvin cycle. Now Snehangshu asked a question. Snehangshu asked that why is the name of Kelvin only used for the cycle and not all the three scientists? The answer to your question that it was only because when we suppose, see Snehangshu, I'll tell you here. So this is some poetry I do sometimes, don't see. Okay, so Snehangshu, to answer your question, suppose for example, you are a research professor. You're a professor or a senior research scientist. Senior research scientist. Okay. Now, you are Snehangshu. Now, there is me, Abhishek Datta, and then there is Pritika. Pritika and I work under you. Okay. Now, it was Pritika's idea. It was Pritika's idea that, okay, we can do this experiment this way. But she couldn't execute it alone. She took my help. And both of us worked in the laboratory and submitted our work to you. You have previous works based on photosynthesis, but this is our new work. You checked our work and you said that, okay, fine, your work is fine, I will correspond this to a journal, a scientific journal. Maybe Nature, maybe Science, these are journals. Okay, you corresponded them. See, this is our work. When you correspond, you will, in the author, suppose the topic of the research is blah 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 blah. Then at first Pritika's name will be there. Then there will be my name. And then because you have just checked and edited, you will be corresponding author Snehangshu. So it will be Pritika. Now then later on, if this becomes or gets a Nobel Prize, all of us three will get the Nobel Prize. But if the latter it is used as any method in any textbook, it will be used as Pritika's method. In the same way, Kelvin, Benson and Besham. Three of them must have worked together. They must have helped him. Maybe his was the idea was most about him only. So his name is used. Snehangshu, is your doubt clear? Okay. So yeah, today let us do only the historical perspective only. Tomorrow I will take another class of biology. I will text you the time in WhatsApp. Thank you. Till then, bye bye. Tomorrow there will be an extra class of biology.